In a striking shift in the global scientific landscape, researchers in the United States are increasingly seeking refuge in European institutions, spurred by drastic funding cuts and perceived threats to academic freedom under the Trump administration. This trend marks a significant recalibration in the flow of scientific talent, potentially reversing decades of American dominance in research and innovation. As the Trump administration's policies continue to raise alarms among the scientific community, universities across Europe have rolled out welcoming programs aimed at attracting American researchers who have been adversely affected by job losses or have concerns about their work being stifled. Notably, Aix Marseille University in France has introduced a program called Safe Place for Science, which has received an influx of applications from what university officials term scientific asylum seekers. The university is reportedly processing about a dozen applications daily, illustrating the urgency felt by many researchers to relocate. Eric Burton, president of Aix Marseille University, expressed the gravity of the situation, emphasizing that these cuts impact not just individual careers but the foundational principles of scientific inquiry. This program is ultimately linked to indignation to declare what is happening in the United States is not normal, he stated, highlighting the university's commitment to providing hope and opportunities for displaced scientists. The implications of these developments extend far beyond individual job losses. The Trump administration's cuts have targeted critical areas of research, including climate change, public health, and social sciences, which many European leaders view as essential to global scientific progress. Philippe Baptiste, the French Minister of Higher Education and Research, characterized the administration's actions as collective madness, stressing the need for a coordinated response from the global scientific community. He underscored the interconnectedness of research, stating, "They are making decisions that call into question whole swaths of research, not just in the United States but the world." As European institutions mobilize to fill the void left by the U.S. cuts, funding initiatives have begun to take shape. In France, a cancer research foundation has pledged €3.5 million to support American cancer researchers, while other universities have announced positions for those whose projects have been halted. This influx of resources reflects a strategic effort to maintain and enhance the quality of research within Europe, with a particular focus on areas like climate science and gender studies. The response has also been echoed across the continent, with leaders in the Netherlands and Germany advocating for increased investment in science to attract displaced researchers. Ulrike Melmendier, a prominent economist in Germany, described the current situation as a unique opportunity for Europe to bolster its scientific community and attract talent from the U.S. The potential for a long-term shift in the scientific landscape cannot be understated. As more American researchers make the move to Europe, there is a palpable concern that the United States could risk losing a generation of scientific expertise. Yasmin Belkade, president of the Pasteur Institute in Paris, warned of the long-term consequences, stating, "We could lose a generation of science, a generation of scientists, something that we cannot recover from." As the world watches this unfolding situation, the conversation around academic freedom and the funding of scientific research is likely to intensify. With European institutions actively courting American researchers, the coming years may witness a significant reshaping of the global scientific community, challenging the traditional power dynamics that have long favored the United States. The outcome of this migration not only has implications for research quality and innovation, but also poses questions about the future of scientific collaboration on a global scale. In conclusion, the trend of U.S. researchers seeking opportunities abroad raises critical questions about the future of science and academic freedom in a rapidly changing geopolitical landscape. As European universities extend their outreach and resources, the potential for revitalized scientific inquiry in Europe appears promising. However, the significance of these developments will depend on the ability of American institutions to adapt to changing circumstances and uphold the values of academic freedom and robust funding in the wake of political turbulence. In a groundbreaking study published in the Proceedings of the National Academy of Sciences, Researchers at the Institute of Science Tokyo have unveiled a novel mechanism of intercellular communication that could have profound implications for stem cell biology and regenerative medicine. Led by Professor Takanori Takebe, the team's work demonstrates how messenger RNA-mRNA can be transferred between different types of stem cells, potentially influencing cell fate and differentiation. Cell communication is a fundamental process in biological systems traditionally understood through well-established mechanisms. However, the role of RNA in this communication has been less explored until recent years. The study highlights an intriguing pathway for mRNA transfer via tunneling nanotubes, thin tubular structures that connect neighboring cells. While previous research primarily focused on extracellular vesicles for RNA transmission, this new discovery sheds light on a more direct method of intercellular communication. To investigate this phenomenon, the research team employed a co-culture system. Pairing mouse embryonic stem cells mescs with human primed pluripotent stem cells HPSCs. This setup allowed the researchers to track the movement of mRNA based on the distinct genetic sequences of the two species. When we started this co-culture for a different purpose, we almost serendipitously found this unexpected mRNA transfer phenomenon, Takebe noted. The results revealed that mRNA coding for critical functions such as transcription, translation, and stress response was transferred from mescs to HPSCs, occurring through the tunneling nanotubes. The implications of this mRNA transfer are significant. The study found that the transferred mRNA could revert primed HPSCs back to a naive state, an earlier stage in their developmental path. This conversion suggests that mRNA exchange between different mammalian stem cells is not merely a passive process, but one that actively influences the fate of the recipient cells. The identification of key transcription factors involved in this process further emphasizes its biological significance. First author Yoneyama articulated the importance of these findings, stating, "This study provides insights into a novel mechanism of intercellular communication, illustrating how cell populations coordinate and coexist with their surrounding environment." Takebe expressed hope that the research could pave the way for new technologies in cell fate control, eliminating the need for artificial gene introduction or chemical treatments, which could transform therapeutic strategies in regenerative medicine. Looking ahead, the research team is eager to delve deeper into the complexities of intercellular communication and its potential applications. As they explore this uncharted territory, their findings may lead to innovative approaches in stem cell therapy and regenerative medicine, offering new avenues for treating a range of diseases and conditions. In a separate but equally significant development, the landscape of mRNA research is being influenced by shifting political tides. With the recent appointment of Robert F. Kennedy Jr. as Secretary of Health and Human Services, concerns are growing regarding the future of mRNA technologies. The Trump administration's Operation Warp Speed, which expedited the development of COVID-19 mRNA vaccines, is now facing scrutiny as vaccine skepticism gains traction. This shift could hinder the momentum of mRNA advancements at a critical time when researchers are making groundbreaking strides in the field. As the Institute of Science Tokyo continues to unlock the mysteries of mRNA transfer, the broader implications of these discoveries will likely resonate across various scientific domains. The intersection of policy and scientific innovation remains a crucial area to watch, as developments in mRNA research could shape the future of medicine, influencing everything from vaccine development to regenerative therapies. As we stand on the brink of these advancements, the need for supportive policies that foster scientific exploration has never been more apparent. In a groundbreaking advancement in cancer research, the Weill Family Foundation has made a transformative commitment of $50 million to establish the Weill Cancer Hub East, a collaborative initiative aiming to revolutionize cancer treatment through an innovative exploration of the interplay between nutrition, metabolism, and immunotherapy. Announced on March 27, 2025, this partnership unites four prestigious institutions: Princeton University, the Rockefeller University, Weill Cornell Medicine, and the Ludwig Institute for Cancer Research. Immunotherapy has emerged as a promising treatment modality leveraging the body's immune system to combat cancer. However, its effectiveness can vary significantly among individuals and different cancer types. The Weill Cancer Hub East seeks to address this variability by fostering cross-disciplinary collaboration that integrates expertise from diverse fields including cancer biology, clinical trials, immunology, and nutrition. This multidisciplinary approach is poised to yield groundbreaking insights into how dietary factors and gut microbiomes influence the success of immunotherapy. The establishment of the hub is a notable achievement for the Weill Family Foundation led by philanthropists Joan and Sanford I. Weill, who have a long history of supporting advancements in science and medicine. The Weill Cancer Hub East will offer doctors and scientists a tremendous opportunity to revolutionize the treatment of cancer, said Sandy Weill. The total financial commitment from the foundation and the partner institutions exceeds $125 million, reflecting a collective dedication to pushing the boundaries of cancer research. Over the next decade, the hub will conduct comprehensive studies into the relationship between solid tumors and their surrounding environments, investigating how factors such as nutrition and microbial composition can enhance the immune response to cancer treatment. One area of particular interest is the potential impact of GLP-1 agonists, a class of drugs typically used to manage diabetes and obesity, on cancer progression and treatment outcomes. Dr. Robert A. Harrington, Dean of Weill Cornell Medicine, emphasized the hub's mission to answer critical scientific questions about immunotherapy's efficacy. By convening world-class institutions with leading investigators, the Weill Cancer Hub East seeks to discover new ways that nutrition and gut microbes can improve the immune response to the disease, he stated. The hub's research will utilize cutting-edge techniques in metabolomics and artificial intelligence to further understand how metabolic processes affect the immune system's ability to control cancer. Christopher L. Eisgruber, president of Princeton University, highlighted the importance of interdisciplinary collaboration in driving transformative scientific advances. In addition to providing seed funding for innovative research projects, the hub aims to support a range of clinical trials examining the effects of metabolic modulation on cancer treatments. Through its structure, the initiative promises to adapt to evolving research landscapes while fostering the development of the next generation of cancer researchers. The Weill Cancer Hub East represents the second collaborative venture initiated by the Weill Family Foundation. Its predecessor, the Weill NeuroHub, established in 2019, similarly focuses on interdisciplinary collaboration to accelerate advancements in neurological and psychiatric diseases. The Weill family's philanthropic efforts have contributed over $1 billion to various causes globally, reinforcing their commitment to advancing scientific research and patient care. As the Weill Cancer Hub East embarks on its ambitious journey, the potential for meaningful breakthroughs in cancer treatment looms large. The collaborative efforts of these esteemed institutions may lead to enhanced therapeutic strategies that significantly improve patient outcomes, marking a pivotal moment in the fight against cancer. Looking ahead, the integration of nutrition and metabolism into cancer treatment paradigms could reshape the landscape of oncology, paving the way for a future where personalized, effective therapies become the norm. As artificial intelligence continues to transform various fields, its integration into the realm of data analysis and scientific research is becoming increasingly significant. Recent advancements highlight a commitment to enhancing transparency and fostering knowledge exchange, particularly through innovative tools designed to streamline collaboration among researchers. Notably, the introduction of the AI assistant known as git-bob and enhancements to the Web of Science Research Assistant are pivotal in this evolving landscape. Developed by Robert Haase and his team, git-bob is a generative AI tool that integrates with GitHub and GitLab, allowing for a collaborative environment where domain specialists can engage with an AI assistant. This system addresses a growing concern in scientific publishing: the challenge of distinguishing between human-generated and AI-generated content. As Haase points out, the prompts sent to LLMs are commonly not conserved, making it difficult to differentiate contributions. git-bob stands out by allowing multiple users to interact within a single communication thread, facilitating knowledge exchange and transparency in code generation. The interactions are documented, enabling researchers to follow discussions and code modifications over time. The open-source nature of git-bob further enhances its utility, allowing developers to customize the AI's responses and integrate it with various LLM service providers. This flexibility not only promotes transparency but also encourages a collaborative atmosphere where scientists can work together more effectively, ultimately leading to more robust scientific outputs. In parallel, Clarivate has also made significant strides with its Web of Science Research Assistant. Launched in September 2024, the tool leverages generative AI capabilities to enhance researchers' interactions with the Web of Science core collection data. Philip Ryman, Director of Product Management at Clarivate, emphasizes the importance of user feedback in refining the tool, stating, "By listening to feedback and analyzing customer needs, we have delivered impactful features that help our users research smarter." Recent updates to the Web of Science Research Assistant include editable queries that allow users to refine their searches more efficiently and structured responses that summarize key themes and conclusions. Additionally, intelligent semantic search capabilities expand the scope of search results, ensuring users capture all relevant literature. These enhancements are designed to empower researchers to navigate the vast landscape of academic literature with greater ease and efficiency. Moreover, the assistant now offers high-value visualizations that help users understand the connections between researchers and their work. Features such as co-author maps and publication trend graphs provide insights into collaborative relationships and emerging research areas, further supporting researchers in their projects. The dual advancements represented by git-bob and the Web of Science Research Assistant mark a significant step towards making scientific research more transparent and collaborative. As these tools become more integrated into the research process, they promise to enhance the efficiency and efficacy of scientific inquiry. However, with the rapid evolution of AI technologies, it is essential for the academic community to remain vigilant about ethical considerations and the potential implications of AI on research integrity. Looking ahead, the continued development of AI-assisted tools in research will likely yield even more collaborative frameworks, fostering innovation and driving discoveries across disciplines. As researchers embrace these technologies, the challenge will be to balance the benefits of AI with the need for transparency and accountability in scientific practices. The future of research may well hinge on how effectively the academic community navigates these transformative changes. In a groundbreaking development for nuclear energy, China is on track to construct the world's first fusion-fission hybrid power plant by 2030. Named the Xinghuo reactor, this ambitious project aims to generate 100 megawatts of continuous electricity, a feat that could position China at the forefront of nuclear innovation and energy production. The Xinghuo reactor represents a collaboration between the state-owned China Nuclear Industry 23 Construction Corporation and Lionovation Superconductor, a company that emerged from Lionovation Optoelectronics. Located on Yaohu Science Island in Nanchang, Jiangxi Province, the facility is set to undergo a comprehensive environmental impact assessment which will evaluate its effects on air, water, noise, and ecological systems. The project, which has a budget of approximately $2.76 billion (20 billion yuan), officially entered its first phase with the issuance of a public tender for the environmental study. China's strategic ambition for the Xinghuo plant is underscored by its goal of achieving a Q value, a measure of energy gain, exceeding 30. In comparison, the International Thermonuclear Experimental Reactor ITER in France is targeting a Q value above 10, while the U.S. National Ignition Facility recorded a Q of 1.5 in 2022. This high Q value indicates that the energy output from the fusion reaction will significantly surpass the energy input required to sustain it. The innovative design of the Xinghuo reactor combines the processes of fusion and fission. Fusion, the process that powers the sun, involves the merging of light atomic nuclei, while fission breaks apart heavy atomic nuclei like uranium. By harnessing the high-energy neutrons produced during fusion reactions to trigger fission in surrounding materials, the plant aims to enhance energy output and reduce nuclear waste. This hybrid approach not only promises a more efficient energy solution but also redefines the potential of nuclear technology. China's efforts in nuclear innovation are timely as the nation seeks to bolster its energy security and transition away from reliance on fossil fuels. The hybrid plant is expected to be operational by 2030, aligning with the country's broader energy goals. If successful, it would place China years ahead of other countries in the race for advanced nuclear technology, potentially reshaping the global energy landscape. As the project progresses, its implications extend beyond energy generation. The success of the Xinghuo reactor could accelerate the development of other large-scale nuclear projects, such as the China Fusion Engineering Test Reactor, which is slated for the mid-2030s. This advancement could position China as a leader in the nuclear sector, fostering further research and innovation in fusion technology. In conclusion, the Xinghuo reactor stands as a testament to China's commitment to pioneering new frontiers in energy generation. As the world grapples with the pressing challenges of climate change and sustainable energy demands, China's potential to lead in fusion-fission technology could not only change the dynamics of global energy production but also pave the way for a cleaner, more sustainable future. The coming years will be crucial in determining whether this ambitious vision can be realized and how it will influence the global energy landscape.